Alrighty, hello guys. Welcome to Module 2 or Week 2 lecture. Give me 1 second, I'm having some issues with my computer. Alright, here we go. So this is Module 2, Parasitology, Virology, Mycology. And in this lecture we're going to be covering Chapters 3 and 4. Chapters 3 and 4 include the platyhelminths. So this first lecture, we're going to cover Chapter 3, which is the Cestota, and then in Chapter 4, we're going to be covering the Digina, but they both fall under the same phylum, platyhelminths or flatworms. So again, here, your learning objectives for you guys to review. All right, so an introduction, classification. Platyhelminths, this is the phylum, this is the flatworm um, phylum. Hold on 1 second, my printer keeps going off. All right, I think we're done printing now, so I should be good to record again. Okay, so again, this is Chapter 3, Cestota, and Cestota falls under the phylum of platyhelminths, also known as flatwormed. These are obviously flattened worms, but they have solid bodies with no body cavity. We have two classes in the phylum of platyhelminths. We have the Cestota, which are also known as tapeworms, and then we have the Digina, which are known as the trematodes and flukes. Both of these uh, classes are hermaphroditic, which means that they contain both the male and female parts in the same individual. General body structure, these flatworms have a scolex or scoliosis. This is on the anterior end of the tapeworm. It attaches to the wall of an intestine of a host by means of suckers or hooks. So the scolex is what the helminth is going to use to attach to our intestines. And then we also these worms also have what is known as a proglottid, and this is just a segment of the tapeworm, and each proglottid contains male and female reproductive organs. So the first tapeworm that we're going to discuss is Hymenelepis nana or nana. This is also known as the dwarf tapeworm. This organism is found worldwide. It's common in the southeastern states of the United States. The adult worms are going to live in the intestines, and how humans are infected is we're going to accidentally ingest an egg from contaminated food or water, or from hand to mouth. And then it's common for people to do an auto reinfection. So if somebody is essentially excreting the eggs and the feces, they could possibly do a hand to mouth fecal oral route and reinfect themselves. The diagnostic stage is going to be the egg, and we have a picture of that on the next slide, so we'll just wait to go over the egg whenever we get there. Major pathology and symptoms. So light infection is asymptomatic, so we're not going to see symptoms. A heavy infection could include intestinal enteritis, abdominal pain, diarrhea, headaches, dizziness, and anorexia. It's usually common to have multiple adult worms present whenever we have a current infection. The dwarf tapeworm requires no intermediate host. A house mouse is common definitive host. Fleas and beetles can serve as transport hosts. So we could possibly get an infection from mouse turds that are infected with the egg. So we'll just go to the next slide and have a look at the egg here. This is the diagnostic stage. So most of the time in parasitology, especially, but in the lab, it's small details, one simple detail that could help lead us to a diagnosis. So here we see the diagnostic stage or the egg. So with this egg, you see kind of a colorless shell here. We have filament emerging from the polar thickenings. So I don't you can kind of see those filaments here emerging. We have hooklets inside the egg right here. There's usually three pairs of those hooklets in the egg, and then you kind of see it's you've got an internal structure and then an external structure. That external structure is a colorless shell. So this is a Hymenalepis nana egg. The next organisms or parasites we're going to cover is the just simply known as tapeworms. We have the Tania saginata and the Tania solium. It's very, very important that you guys are able to recognize that saginata is beef, solium is pork. And what that means essentially is that if we eat raw or undercooked beef, we could possibly be infected with a beef tapeworm. If we eat raw or undercooked pork, then we could be infected with the pork tapeworm. So Tania solium is the pork tapeworm. The diagnostic stage is we could see eggs and feces. We're also going to be looking at intact gravid proglottids, usually in pairs or short chains in the Tania solium and usually singly in the Tania saginata. And then we could do radiographic computed tomography or CT scan, magnetic resonance imaging demonstration of cysticerus in the tissue that accompanies the symptoms. Symptoms, most infected people are asymptomatic, but they can experience abdominal pain, diarrhea, and weight loss. Eosinophilia is moderate, so we're not going to have a large number of eosinophils present. Humans can serve as accidental or intermediate hosts for Taenia solium. Uh, in the eggs of Taenia solium, they're going to be accidentally ingested and released from a ploglottid in the intestinal tract. The eggs will hatch in the intestine and the larvae will migrate to form cysterus bladders in any organ or nervous tissue. So let's have a look at the diagnostic stage. Like, we need to talk about distribution. Taenia saginata it occurs in countries where beef is eaten raw or insufficiently cooked. It's found in the southern, southwestern United States areas. Tania solium is where pork is eaten raw, and it's not as common in the United States. Excuse me. So we most often hear about Tania saginata infections happening in the United States. So here we have a picture of the adult Tania saginata worm or the beef worm. You can see here that it's rather long, coily worm. We've got areas of proglottids here, and then we have the scolex, which is the part that adheres to our intestinal walls. We have the proglottid section here with pores, uterine branches with eggs. So usually counting the uterine branches is partially how we could diagnose which specific worm we have. Here we have a picture of the egg. You could see that we have internal embryo, radial striations on yellow-brown embryophore. So you see we have these little striations here in the outer side of the egg. And then we got three pairs of hooklets. They're a little harder to see in here, but there's going to be pairs of hooklets here inside of these eggs. So it's important that we're able to identify this egg as a tapered egg. And then from there, we will go into the uterine branch comparison to figure out specifically if we have a solium or saginata egg. But it's generally, if we ask patients if they've eaten undercooked beef or undercooked pork, we could pretty much tell which organism we're going to have. You can see here that the Taenia solium, the proglottids are a little bit lessened, thicker, and more spaced out. And then with the saginata uterine branches, there are quite a few more, and they're a little bit less spaced out in between them. So it's just something to remember here that saginata is a beef tapeworm. It has, in its gravid proglottid, it has quite a few more uterine branches than we will see in the Taenia solium, which is the pork tapeworm, which has fewer branches, but the eggs generally look the same here. So we want to focus on being able to recognize an egg. The next parasite that we're going to discuss is Diphyllobothrium latum, also known as the broadfish tapeworm. So how we would diagnose the Diphyllobothrium latum again is we're going to recover an egg and feces. We could also see evacuated proglottids in chains ranging from a few inches to several feet. Here, typically, we would mostly just look for an egg because there's generally only one adult present, so it's a little bit harder to get recover an adult worm. So again, the diagnostic stage here is going to be the egg, which we'll see on the next slide. Disease name would be Diphyllobothriasis, or a fish tapeworm infection, or broadfish tapeworm infection. Major pathology and symptoms would be an intestinal obstruction because an adult can grow up to 20 meters long. Patients are going to experience abdominal pain, a vague digestive symptoms. We could see weight loss and weakness, but about 1% of infected people are going to experience a macrocytic anemia. So that's whenever our red cells are a little bit enlarged. And this is due from a dietary deficiency caused by the tapeworm utilizing all of our dietary vitamin B12. So it's important to know that the Diphyllobothrium latum is associated with vitamin B12 deficiency and macrocytic anemia. These parasites are typically going to be found in regions where freshwater fish are a common part of the diet or raw fish are eaten. So in the United States, we're going to see infections with Diphyllobothrium latum more common in the Great Lakes regions or Alaska. We'll also see it in Chile, Argentina, Central Africa, and parts of Asia. Patients of the Baltic region or Finnish people are going to be most of our infectious, where most of our infections are going to occur in Europe. So let's just have a look here at the egg. So here we see the egg of Diphyllobothrium latum. We can take a closer look and see that we have an operculum at one of the polar ends of the egg here, and then at the other end we have a terminal knob. Trying to circle it here. So we have a terminal knob here. Then we have underdeveloped embryo inside of the egg. So this is an egg for Diphyllobothrium latum, so it's just important to be able to note that we have that terminal end and that operculum. The next parasite that we're going to cover is Echinococcus granulosus, or the hydatid tapeworm. Method of diagnostic, we could do serological testing for this parasite, indirect hemagglutination, or ELISA testing. We're going to look for presence of scolices, broad capsules, hydatid sand, or daughter cysts in the hydatid cyst fluid. Generally, we're going to do a biopsy, but it's not recommended because a biopsy can leak cyst fluid and cause anaphylaxis. We can detect the organism from X-ray examination, ultrasound, or CT detections of cyst mass in the organs. Sorry. Disease names are going to be hydatid cyst, hydatid disease, echinococcus. Major pathology and symptoms. Symptoms are going to vary according to location and size of the cyst. Expanding cysts cause pressure, necrosis of surrounding tissue. The most common site is going to be in the liver. We're not going to see any symptoms until the cyst gets larger, so it's going to be nearly a year after ingestion of the egg, and we're going to see jaundice or portal hypertension. In lung infections, we're not going to see symptoms again until the cyst is large. Then the patient may experience coughing, shortness of breath, and chest pain. Whenever a cyst can grow or rupture, it can result in the death of a patient. Again, this is because it causes anaphylaxis. So anaphylactic shock may occur, and if the cyst ruptures, may occur during a biopsy. We also may see eosinophilia and a bronchospasm. Treatment for this parasite is going to be a surgical removal of the cyst. And then human infections occur as zoonoses, primarily in sheep raising areas where domestic dogs are used in herding. So in sheep raising areas, chiefly among the Navajo people, Utah and Alaska is where we're going to see infections of Echinococcus granulosis. Here's a picture of the hydatid cyst and the embryonated eggs. So here we see a picture of the adult, and it's a little bit shorter of an organism. You can see it has that sucker at the end that it's going to use to adhere, and then that parasite is actually going to create this hydatid cyst, and infection occurs from ingesting an egg. So let's just have a look here at the egg. So that's going to do it for chapter 3 of Cestota. I think it's very important for you guys to take the time to review your book and do the post testing at the back of the chapter. Make sure that you go through and learn the definitions in the glossary at the beginning of the chapter, because they're going to help you really understand a lot more in depth of what is happening with these parasites and the parts of these parasites. The next chapters we're gonna cover is Digina. Uh, Digina are commonly called flukes because they belong to the phylum Platelmints, along with the Cestota. These parasitic infections are gonna inhabit the intestines or tissue of humans. So before I get into too much detail, I think we should go over the learning objectives here. So again, here are the learning objectives. These um, presentations are available for you guys to download. So if you want to go through and download these PowerPoints, that you could get these learning objectives down, that's available for you to do. So we already covered the classifications, so let's get into a little bit of the body structure. So the Digina class are flattened and non-segmented. They're leaf-shaped helminths. Trematodes vary in length from a few millimeters to several centimeters. Adult flukes have two cup-shaped muscular suckers or astab as acetabulula. I've never heard of that term, but they have these oral suckers, and that's generally what they're going to be referred to in parasitology, is just that they have suckers. We could have two different types of parasitic flukes that could be present in humans. One type lives in the intestine and other host organs, so the liver and lungs. So we have blood flukes and intestinal flukes. So schistosoma are what we're going to be referring to as our blood flukes, and these flukes are also hermaphroditic, which means that contain um, they contain both the male and female reproductive organs. So let's just jump right into the organisms here. The first group of organisms we're going to cover, or parasites, are Fasciolapis buski, which is also known as a large intestinal fluke, or Fasciola hepatica, which is known as a sheep liver uh, fluke. The diagnostic stage here is going to be, again, the egg, recovering the egg, and the egg is similar for both species, so we'll get into that into the next slide. So we're going to recover the egg from feces. Eggs of these two species are too similar to differentiate. Species diagnosis depend on clinical symptoms, travel history, and recover of adult Fasciolapis uh, organism. Fasciolapis buski infection disease name could be listed as fasciolapiasis, and then Fasciola hepatica is just known as a sheep liver rot. So symptoms with Fasciolapis buski include bowel mucosal ulcers and hypersecretion and occasional hemorrhage around worm attachment site. Heavier infections are gonna patients are gonna experience pain, nausea, mucoid diarrhea, anemia, intestinal obstructions, and malabsorption. Generalized edema, which is retention of fluids, and marked eosinophilia. Death in a patient can occur with heavy infections. With fasciola uh, hepatacea, symptoms are from mechanical irritation, toxic worm metabolites, and mechanical obstruction. Migration of larvae can cause local irritation. Patients are going to experience fever, hepatomegaly, eosinophilia in humans from endemic areas such as clinical diagnosis. Adult flukes in the bile duct induce portal cirrhosis to heavy infections. So patients are going to experience jaundice, duct obstruction, diarrhea, and anemia. It's important here to know that fasciola hep, 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 hepatisa, it's known as the sheep liver fluke. So those of you again, we're going to refer back to medical terminology. The hep means liver. So this is going to be known as the sheep liver fluke, and the symptoms are going to be largely based on infection or obstruction of the liver. So again, here we're going to see jaundice, bile duct obstruction. We're going to see hepatomegaly, which is an enlargement of the liver. So that's why it's important to know that it's a sheep liver fluke. Fasciolopus buski is most prevalent in China, Vietnam, Thailand, Indonesia, Malaysia, and the Indian subcontinent. Fasciola hepatica, it has cosmopolitan distribution in sheep raising and cattle raising countries, but it is uncommon in the United States. Here's just a picture of the egg. We see again we have a little operculum end of the egg and underdeveloped metastasium inside here. So it's just important to be able to mostly be able to recognize the symptoms, travel history, and recovery of the adults. Okay, this one's a little bit harder to pronounce. Chlorochlorocus sinensis, or the Oriental or Chinese liver fluke. So here, right in the name, Chinese liver fluke, Chlorosis sinensis. So method of diagnosis, we're going to recover and identify eggs in the feces or biliary drainage from entero test capsule. Differentiation of species is difficult. We could also perform radiographic studies. Disease name is chlorocalciasis. Pathology and symptoms, so light infections, patients can be asymptomatic. Other patients with heavier infection can experience jaundice from bile duct pathology. Hyperplastic changes may occur. Hepatomegaly may develop with tenderness in right upper quadrant. Other symptoms include abdominal pain, diarrhea, and anorexia. Some patients who have a chronic infection may develop hepatic complications, sometimes pancreatitis, bile duct stones. Uh, this parasite is found in the Far East, typically found in Asia, which is probably why it's called the Chinese liver fluke. Eggs are passed intermittently, therefore performing sequential stool examinations is recommended. So what this means is you're gonna collect more than one stool sample to try to collect an egg or diagnose this organism. Here's a picture of the egg. So here, the patient's going to have a thickened rim around the operculum, also known as shoulders. You're going to see development casetum inside of the organism. You could see these little developments inside of there. And then we're going to see a really tiny small knob at the end here. You see that little knob present at the end of the egg. So this is the clonorosensis sinensis egg. These little shoulders are, and just the structure of the egg, the way it looks, is going to be how you diagnose this egg, and it's really kind of, standalone. You could. All right, the next parasite we're going to talk about is Heterophes, and Metagonomus yokagawi. You notice that some of these names are a little bit harder to pronounce, and they kind of sound Chinese, but that's because that's where some of these infections are found, especially since in China and Asia, it's a little bit more common to eat raw and undercooked fish and snails and so on and so forth. So the method of diagnosis for these parasites we're gonna recover and identify eggs and feces or duodenal drainage. Differentiation is difficult. Some may lack the knob at the shell end, opposite of operculum. Just like the last organism we saw, some of them may lack that terminal knob. Disease names, it's going to be heterophiasis, or metagonomiasis. Um, oh, that's metagonomous. Sorry, some of these things are just now coming back to me. It's been a few years since I've lectured parasitology. The infection is going to be asymptomatic. Heavy infections may include a chronic mucoid, may induce chronic mucoid diarrhea and abdominal pain. Worm invasion may produce eggs that travel to the heart and brain, causing symptoms of granulomas. Distribution: Heterophes occurs in the Near East, Far East, and parts of Africa. Metagonomus yogowawi is found in Asia or Siberia. These parasites are primarily found in dogs, cats, and other fish-eating mammals. So human infection is a zoonotic infection. So we're not, it's not very common for humans to get infected. Eggs can travel to the tissues and cause granulomatous or tissue disorders. And Heterophes has a third sucker around the genital opening. Let's just have a look here at the egg. So you see here, there's no terminal nodule at the end of this egg, but we do have that shoulders or operculum in the front. So this is just in the egg of these two organisms. So make sure whenever you're trying to differentiate Clonorocensis and Heterophes, and Metagonomus, that you're noticing that lack of that end. But the shoulders aren't on this organism aren't as pronounced as the last organism. All right, the next parasite we're going to talk about is Paragonomus westermani, also known as the Oriental lung fluke. How we're going to diagnose this uh, parasite is we're going to recover and identify eggs and bloody sputum. Sputum appears to be contain rusty iron fillings, or in feces, we're going to see if the eggs are swallowed, we may see them in the feces. We could also take a radiograph of the lungs and show patchy infiltrate with nodular cystic shadows or calcification. The disease name is paragonomiasis. Um, Most of the infections are going to come from eating snails or freshwater crabs or crayfish. And I highly recommend that you read the book and get a lot more detail on these parasites. Major pathology and symptoms, we're going to see chronic chest pain, cough, tinged sputum, so rusty looking sputum. Adults are present in fibrous cysts, and eggs pass through cysts and rupture into the bronchioles, causing a cough. We could also see a cerebral paragonomiasis infection, and these are usually asymptomatic infections. Distribution is going to be again the Far East, Africa, and South America. There's at least 8 other species of Paragonomus that can be also infectious for humans. So here again, we're just going to want to take a quick look at the egg. See, it's a lot more thick than the last couple eggs, but we still have that little nodular end, or the operculum end here. All right, the next parasite that we're going to talk about are schistosoma species. So these are, there's a couple of different species, but these are generally known as the blood flukes. So method of diagnosis, we're going to see schistosoma mansoni and schistosoma japonicium, where we could recover eggs and feces or a rectal biopsy. We have schistosoma hematobiome, and here we could recover eggs in urine. And this information can be found in your book, and I highly recommend that you go through each specific species and see where you're gonna recover eggs. But here, in general terms, the diagnostic stage that we're gonna look for is the egg. Disease names can be swamp fever, schistosomiasis. Um, these diseases mainly affect children and adults in endemic areas. The initial reaction may be dermatitis or from cesareal penetration. Acute phase of heavy first infection present. Patients can experience typhoid fever-like symptoms, so fever, cough, malaise, hepatosplenomegaly. We could see cirrhosis of the liver, bloody diarrhea, bowel obstructions, hypertensions. Eosinophilia is going to be present here. And then we could see nephrotic syndrome occur in schistosoma mansoni and schistosoma hematobiome. Schistosoma mansoni is found in Africa and South and Central America. We could also see this in the Caribbean. Schistosoma japonicum is found in the Far East, and Schistosoma hematobiome is found in Africa and the Middle East. So schistosoma or blood flukes are going to rank second behind malaria as a cause of serious worldwide morbidity and mortality, and it affects more than 200 million people a year. Schistosoma mansoni infections are found in large urban areas such as New York and Chicago, with immigrant populations from the Caribbean islands. So let's just have a look at the next couple of slides because it's a little bit easier to think about these organisms whenever you could see the actual egg. So here we have a schistosoma mansoni egg, and we see that large lateral spine on the end. And then we have schistosoma hematobiome. This is from a urine sample here, and then we see that you have that large terminal spine at the end here. And then we have schistosoma japonicum. This one also could have a lateral spine, but it's a little bit harder to see. And you can see in this slide that it's not very present here. But again, it's important to know that you could either get a stool sample or a urine sample depending on what species you have. And that's it. I highly recommend again that you guys go through and do the post test in your book. Make sure you cover your homework and review the terms at the beginning of the lecture. I mean chapter.